So first of all, good morning, everybody. I'm really happy to be here to present my work about affinity-capture mass spectrometry for the discovery of receptor-targeting peptides in animal venoms. So this is a classical presentation, in fact, classical introduction when we speak about animal venoms. You know that they contain a lot of bioactive peptides that are very promising as leads for drug discovery, but also for therapeutic development. But the discovery process is still limited, not only because venoms are very complex, we have tens, hundreds, thousands of compounds in these venoms, but also because of the low throughput of the traditional screening methods like bioassay guided fractionation, for example. And there is still a pressing need for efficient, high throughput methodology to discover peptides that bind to receptors directly from crude venoms. Why did we choose to work with membrane receptors? Because they play critical roles, of course, in numerous cellular pathways, but also in many diseases. We have three main families of membrane receptors: the ionotropic receptors, also known as ligand-gated ion channels, the metabotropic receptors, GPCRs, or also the enzyme-like receptors. And from here, why do we need to discover, to identify selective ligand for membrane receptors? Because of course we want new drugs, but we want new drugs to improve the therapeutical precision and minimize the side effects. It's very important for us. And we want also to discover and to develop new innovative pharmacological tools and powerful contrast reagent for imaging, like we can do with a chlorotoxin, for example. And why doing it by mass spectrometry? Because it's fast, it's sensitive, it's selective, and we don't need any ligand, any labeled ligand to work with mass spectrometry. We see directly the binding of one ligand to a receptor. Our strategy is finally very simple. You just have to incubate your receptors with the mixture of peptides you want to screen, let it incubate for 2 hours here to reach a kind of equilibrium. Then you centrifugate. You recover the supernatant, you collect the supernatant, and in this supernatant, you will have all the compounds that did not bind to your receptors. Then this compound can be identified either by Malditov mass spectrometry if you want to be fast, or by proteomics if you want to deeply characterize these compounds. You can also work on the pellet, and the pellet contains the membrane receptors and the potential ligand that's still bound on it. And you can wash this pellet, acidify the medium to let this compound be released in the medium. And then again, you can collect the supernatant and identify the peptide of the compounds that that are in solution now and that bound to your receptor in the first time. So they are our potential ligand. Concerning the data treatment, the data visualization, we can, of course, directly compare the spectra of the unbound fraction and the bound fraction and see how the compounds are distributed. Or we can also plot the relative proportion of each compound detected in the unbound and the bound fraction to have this kind of histogram. And here you see that the compound number 2 is found not only in the unbound fraction, but also in the bound fraction. You see that it is enriched in the bound fraction, and this compound is possibly a ligand for the receptor we tested here. So we tested the model, the strategy, on a first receptor, which is vasopressin type 2 receptor. It is a G protein coupled receptor, primarily expressed in the renal collecting duct, and it plays a role in fluid balance and blood pressure regulation. We know several ligands for these receptors, vasopressin, of course, which is the endogenous agonist ligand, but it is not selective for the V2 receptor. We have the desmopressin, the tovalpan, and in the world of toxin, we have a very nice ligand for these receptors called mamba-claritin-1 and extracted from the venom of Dendroaspis angusticeps. From an experimental point of view, we worked with human vasopressin type 2 receptors expressed in CHO cells at an expression level of around 11 picomoles per milligram of membrane proteins. So are we able to fish vasopressin from a complex mixture? So we did our mixture with the receptors, with the ligand, and we also added a background of peptide made of bovine cerebralbumin that has been digested by trypsin, and we have then a mixture of around 100 peptides. So you have the reference spectrum here, and in this spectrum, you can identify the signal of the AVP. This is the result. You have the unbound fraction that contains a lot of peptides coming from the BSA. We still have an excess of vasopressin, but in the bound fraction, as you see, you have quite exclusively the vasopressin that is in this fraction. All the small signals are coming from compounds that are released from the membranes during the acidification state. So we are indeed able to selectively capture vasopressin from a complex mixture, but what about animal toxins? As I told you, we have a very nice ligand for these receptors, which is membraquaretin-1. So we make a fractionation of the Novaspis angusticeps, detect the fraction where is the membraquaretin-1 was eluted, and we use this fraction to make the affinity capture. So same receptors, membraquaretin-1, and the background is then made of the other toxins that are in the fraction. These are the results. So you see that the unbound fraction is quite similar to the reference spectrum, but the signal of MQ1 was decreased a lot, and MQ1 is found in the bound fraction, which is a good thing. But we have also a lot of additional signals everywhere. These signals are mainly coming from membrane-release compounds, as I saw in the previous slide. But we still have this compound here. And this compound comes from the fraction. It's not coming from the membranes themselves. And it has been identified as a toxin F8, which is not a ligand for N-acetylcholine receptors. At least it's not described as a ligand, but it's a cytotoxic agent against adenocarcinoma cells. At this time, we don't know if it's non-specific interaction, if it's a membrane adsorption, or if it's a real true affinity. It has to be checked. We wanted to test also to propose a kind of competitive binding monitored by mass spectrometry. And the idea was to try to confirm that vasopressin competes with membraquaretin-1 for the same binding site. And as you see, the percentage on MQ1 bound to the receptor decreases when the concentration of vasopressin increases. So we have a displacement and a competitive binding. So here it seems to say that vasopressin shares or overlap the same binding site as membraquaretin as it was expected. We did the same with the second toxin F8. And as you see, only a very small proportion of this toxin is bound in the beginning of the experiment, even at very low concentration of vasopressin. It drops sharply up to 100%. So we have a complete displacement. And this compound may be a weak binder for the AVP binding site. But we have to remind here that we did this competitive binding experiment by the fraction itself. So we have a lot of compounds inside that compete all together. So we have a competition of AVP and membraquaretin, and potentially this compound, and also this compound and vasopressin. But what is important to realize is that we can visualize several binding events at the same time with this approach. Our second model is the nicotinic acetylcholine receptors. You know that they are pentameric ligand-gated ion channels. They are expressed in muscle, brains, quite everywhere, let's say, promote neuromuscular transmission. They are also involved in Alzheimer and Parkinson's disease and are a major target for insecticides. We know a lot of ligands for these receptors, acetylcholine, which is a non-selective endogenous agonist. We have the nicotine, epipatidine, and in the world of toxin, we have a lot of alpha neurotoxins, alpha bungarotoxin, and we have also a lot of chronotoxin, as discussed in the previous presentation. Experimentally, we use membrane preparation from torpedo marmorata electric organ, which mimics muscle types and acetylcholine receptors, alpha 2, beta, delta gamma. So how are we able to fish ligand for these receptors as well? So we did quite the same experiment with D receptor with alpha J1, which has been presented in the previous talk, and again, the background of peptide. And the results we obtain are the following. In the unbound fraction, we recover mostly the peptides of the background. And in the bound fraction, we have quite exclusively the alpha J1. It's clear that this compound binds to the receptors. And it's also important to note that in the same experiments, we are able here to not only fish a ligand, potentially new, and to make its sequencing in a single step. Of course, between this step and this one, we have to reduce disulfide bond, but it's not so difficult from the fraction because we are enough sensitive with mass spectrometry to detect the component, make MSMS in a second time. Are we able to fish several ligands at the same time? So we reproduced the experiment, but using this time 3 different conotoxins, alpha M1, alpha CN1A, and alpha E1. So you have here the spectrum of the unbound fraction, the spectrum of the bound fraction, and we detect again the signal of the conotoxin. These signals are hard to evaluate here as new ligands, but if we present the data in histograms, then you see that the only 3 signals that are overexpressed in the bound fraction are the one of conotoxin. So we are able indeed to fish several ligands at the same time from a complex mixture. Are we able to fish larger toxins than conotoxin? And for this, we use, of course, neurotoxins and the venom of Naja cautia. This venom has been fractionized, and we have tested several fractions, and I would like to highlight the fraction F36, which is interesting. You have here the unbound fraction with a lot of compounds, and the bound fraction, again, with many compounds. With this toxin, it's very hard to identify the toxin just by the mass, so we had to work with proteomics to identify all these peaks. And we had these results, and we can divide the analysis in 2 cases. The first cases concern the compounds, the toxins that are totally depleted from the initial fraction. And we have, in this case, alpha cobratoxin, as expected, because it's a very nice, very good ligand for N-acetylcholine receptors. And we also got cytotoxin 3 and 1, which are also ligands for N-acetylcholine receptors when they are in their heterodimeric form with cobratoxins. And cobratoxin is also in the fraction, so it makes sense. However, the last one is the cytotoxin 1, which is an isoform from this toxin coming from Naja sumatrana. And this toxin is not a ligand for the receptors, but is described as a pore-forming toxins. So it seems to indicate that indeed we are able to fish ligand for the receptors, whether they interact as monomer or dimers, but also we are fishing compounds that interact with the membranes. We had also this kind of profile, and the first cytotoxin 4 is also a pore-forming toxin. So the reason why it is enriched in the bound fraction is the same as the previous one, because it interacts with the membrane. And the last one is a tryptophan containing weak neurotoxins that is quite well distributed between the 2 fractions. And this toxin is indeed a competitive antagonist of the receptors at a weak micromolar range. So can we do better? Are we able to fish alpha neurotoxin directly from the crude venom itself? So again, we did the same experiment, but instead of using a fraction of venom, we used 1 microgram of the crude venom of Naja cautia. You have again the reference spectrum here with a lot of compounds. You see monomers, dimers, and so on. And these are our results. I will speak about bound fraction, and as you maybe see, you have a lot of peaks everywhere. So again, we cannot identify the toxins directly from their masses. So we used our proteomic approach to identify all these toxins. And we use a more statistical approach this time. we use PCA. And in this PCA, you have 3 groups. These groups are obviously the crude venom analysis, the bond fraction, and the bond fraction. But what is interesting to see here is the tight clustering of each group confirms the acceptable reproducibility of our methodology. We can then visualize the data with a volcanoplot analysis. This volcanoplot show you on the x-axis the fold change of the concentration in log2 basis, and in y-axis you have the log p, which means the statistic significance. Here we compare the bond fraction and the crude venom, which means on the left side of the volcanoplot you have the toxins that are poorly present in the bond fraction, and on the right side you have toxins that are enriched in the bond fraction and then potential ligands. And because we work with proteomics, we are able to identify all these toxins. And what have we inside? We have honestly 2 toxins that can be considered at that time as false positive. These 2 ones, in fact, because they are not linked to membrane pore forming or to binding to an acetylcholine receptor. So we have 2 toxins. We don't know why they are here. We have 2 toxins that are captured due to their activity as pore-forming compounds. We have 7 toxins that are directly linked to an activity with an acetylcholine receptors. We were expecting more, but because we use the crude venom, we saturated probably the receptors, and we could not capture all of the toxins. And what is also important to note is that none of the other compounds detected here in the orange fraction are linked to N-acetylcholine receptors. We have PLA2s, we have SVMPs, we have a lot of different compounds, toxins and enzymes, but not compounds linked to N-acetylcholine receptors. So, take-home message, affinity mass spectrometry is a versatile and high throughput method, which is applicable across many membrane receptors models like GPCRs, ion channels. It's effective for both small peptides and large toxins, from purified to unpurified complex sample. It reduces, of course, the need for extensive purification step for using labeled ligand and even previously known ligand, which can open the door to deorphanization of receptors, because again, we see a direct event of binding of a ligand to its receptor. But negative control, of course, and it's universal, of primary importance to validate the results, and a fast purification, but very fast, is strongly advised to avoid saturation of the tested receptor, because too many binding events could change the data, and we want, of course, to get a better overview of the potential ligand from the venom. I will finish this talk by thanking all the people involved in this work, all the financial support, of course, and a special thanks to Lou Freville, who is a PhD student in the lab and who did mostly the job I have presented today. And I would like to thank you for your attention.